Okay, well, thank you very much for the invitation. This is the title of my talk, and collaborators, especially students who did the work that I'll present, and collaborators Bratislav Misik, who has been collaborating on all of the work that I'll show, and also Louis Collins, who is the person who got me back into Parkinson's disease about 10 years ago. So we all use large data sets, including large MRI databases, to understand, hopefully, disease mechanisms, carry out subtyping, and develop biomarkers. And mostly what we do with these big data is make correlations between brain structures and phenotypes or the like. But as we all know, correlation does not imply causation. Now, I've been inspired by this book, The Book of Why, by Judea Pearl, that I recommend to anyone doing big data science. Pearl is a person who came to the conclusion that actually correlation does imply causation, but that what is not known is what is the architecture of causation. It's complicated and may be hidden, but if there's a correlation, there must be something causing something. And he's developed the whole mathematics of causal inference, but in particular, he advocates the use of causal diagrams, which are derived from path analysis, which I will show you. And also the use, he says, of a symbolic language. And he means, I think, a mathematical language, but symbolic language to express what we want to know. And I take that to mean to make biologically informed models of what we think the causal relationships are, and then use those models with our large data sets to try to understand mechanisms. So I'll show you 4 examples of this from my research. The first one has to do with impulsivity. I think we know what that is. It's a tendency to act without thinking about the consequences of your actions. There have been many studies of impulsivity in brain imaging. One theory is that, let me see if I have. One common theory is that there are 2 mechanisms of impulsivity: defective executive top-down control and increased drive and motivation, which interact together and are associated with different brain systems. However, trying to relate impulsive phenotypes to brain imaging is prone to confounds. People who are impulsive tend to abuse drugs, tend to be overweight, tend to have anxiety and stress, all kinds of things that can themselves change the brain. So how can you understand how a brain develops to become impulsive? So in the last few years, there have been, there are multiple ways to measure impulsivity. There are self-reports. These are validated scales that all measure impulsive trait. There are tasks, like delayed discounting task. There are questionnaires of life events like drug use and speeding propensity in your car. And then there are related phenotypes like body mass index and educational attainment. And what I'm showing here are recent GWASs of all of these traits with very large sample sizes from 100,000 for many of these to 500,000 to 3 million for educational attainment. Many of these come from 23andMe and from other large data sets. So this is the work of Marie Shishikura, who is a PhD student in my lab. And what she did is she took all of these GWASs, which we got from Sandra Sanchez-Roig from UCSD. So you have a GWAS for all of these traits, and then you can see how they relate to each other. So what she did is genomic structural equation modeling of the GWASs. So here are all the GWASs and their associations. And what you can see is that genetically, just like behaviorally, it does split into 2 basic sets, 2 basic factors, and then the second factor splits into 2 more factors. And what you can see, this is the structural equation model, and in SEMs, whenever you have a circle, it means that it's a latent factor, something that you cannot observe directly. And what we find with the structural equation model is that genetically, there are 3 latent factors of impulsivity. One that we call lack of self-control based on its association with these measures, the questionnaires like urgency and poor planning and delayed discounting. one that we call reward drive, which is associated with smoking, drugs, and the like, and one which we call sensation seeking, which is associated with risk tolerance, speeding in your car, and sensation seeking. So genetically, just as in behavior, there appear to be different latent factors of impulsivity. What you can then do is that you can actually do GWAS of these factors. Based on these associations, you can actually now compute GWAS of each of the latent factors. And this is what Marie did. And so we now have GWAS of these three latent factors. And what you can see is that some of the hits are quite interesting. You see this CADM2 and NCAM1. These are genes implicated in brain development, axon guidance, neural progenitor cell development, synaptic plasticity. So it's interesting that the genes implicated in these three latent factors of impulsivity also are genes involved in brain development. So then the next thing Marie did is to take the ABCD cohort, which is 9 and 10-year-olds scanned in the United States, about 5,000 individuals. And so you can compute a polygenic risk score for each of the latent factors and then see their brain associations. And what you can see that they have very different brain associations. So lack of self-control is associated with reduced cortical surface area in the frontal lobes, very consistent with what we know about the mechanisms of self-control. This is entirely driven by the genetics. Then we have the reward drive polygenic score, which is mostly associated with increased cellularity in the basal ganglia. Again, something not too surprising when you know that reward drive is driven by corticostriatal dopaminergic ascending systems. And finally, the sensation-seeking polygenic score is associated actually with increased cortical surface area in various regions. So the conclusion from that, by using genetics, you get around the problem of small sample sizes, and you also get around the problem of the fact that people with impulsivity are going to have exposures in their daily lives that change their brains. The other disease that we recently studied is essential tremor, which is a neurological condition. It's a movement disorder in which people have an action tremor that usually starts in adulthood and responds very well to deep brain stimulation, especially of an area of the thalamus called the VIM, the ventral intermediate nucleus. But there's very little known about the brain abnormalities in essential tremor, which are, first of all, very subtle, and the sample sizes in studies of essential tremor have all been extremely small. So what we thought is that we would take advantage of the fact that there is a recent GWAS of essential tremor. So essential tremor has a very strong familial component. About 50% of people with ET have family history. That's as high as schizophrenia in terms of heritability. And there's a recent GWAS with very large numbers, 7,000 patients and something like 500,000 controls. So very powerful GWAS. And so we took in this particular study, we took the UK Biobank data set and we computed the essential tremor polygenic risk score in 30,000 participants in UK Biobank who did not have essential tremor or a family history of essential tremor. And then we looked for the brain associations of this genetic risk. And the idea here is that genetic risk for Parkinson's disease probably affects the brain in some way, making you vulnerable to some environmental event that later causes and brings out the tremor. This is the work of Miranda Medeiros, who is a PhD student with Guy Rouleau, and Alex Pastor-Bernier, who was a postdoc in my lab. So what we found is that essential tremor polygenic risk score was associated with changes in microstructural anatomy of white matter and gray matter, but especially of a specific tract, the dentato-rubro-thalamic tract, which goes from the cerebellum to the brainstem to the thalamus. And this tract is identified here. So you see that it's mostly these motor structures, motor white matter structures that are implicated here. These people don't have tremor, remember, they just have genetic risk for tremor. And what we show here with the white matter is increased mean diffusivity, which is indicative of microstructural alterations in those white matter tracts. And we can go further and map specifically which tracts are most implicated. And the most affected regions by by MD was this area, the VIM nucleus in the thalamus, which is the site that surgeons have found is the best way to treat essential tremor. So with deep brain stimulation or lesioning, by lesioning this structure, you can actually alleviate the tremor. And this is the structure that is at the center of areas that show microstructural alterations in high genetic risk. So in conclusion, essential tremor genes affect neurodevelopment, I didn't show you that part, of motor control white matter tracts and associated gray matter nuclei. And what we predict then is that there's an unknown environmental trigger that can take a person who's vulnerable to essential tremor and make them express the phenotype. Okay, so how about Parkinson's disease now? So Parkinson's disease, according to Brock, who observed in post-mortem data sets the distribution of pathology, Brock came with the conclusion that Parkinson's disease was a systematic propagating illness, that there was some sort of toxin that starts in the medulla, in the dorsal motor nucleus of the vagus, and in the olfactory bulb, and that spreads throughout the brain. Now, the fact that the olfactory bulb and the gut were the first two sites of disease suggested to Brock that it was probably an environmental toxin that was coming into the body from the outside that caused the disease, and that may well be true. But in any case, what happens is that there's this systematic stereotype progression of the disease, according to Brock, from the medulla up to the brainstem and up into the cerebral cortex. And we now know that the reason for this is that Parkinson's disease is essentially a prion. It is a misfolded protein called alpha-synuclein that in its misfolded state has the ability to misfold other proteins and also to propagate from neuron to neuron transsynaptically. This remained a controversial theory 10 years ago. I think it's mostly accepted now. But we've been using MRI to try to test that theory. So Andrew Vo recently did a study using the Enigma PD data set with 3,000 patients and about 1,000 controls. And Enigma, you probably know what Enigma is, I don't have to describe it. So we had PD patients and at the 4 different stages of the diseases. The stages represent disease severity. And what Andrew found, not surprisingly, it's consistent with what other people have found over the years, that Parkinson's disease is associated with cortical thinning, widespread cortical thinning in a very specific distribution, mostly posterior distribution. And he also found that the amount of cortical thinning correlated with disease stage and also correlated with the presence of cognitive impairment. The other thing Andrew did, which is again something that we've done in our lab that other people have done, is to ask the question, are 2 areas that show atrophy more likely to be interconnected? Because if the disease is caused by some propagating toxin or propagating prion, then connectivity should shape the spatial distribution of brain atrophy. And this is what we found. As I said, other people have been showing this now for 10 years. But basically, the atrophy here, what he shows is that the atrophy of your neighbors correlates with your atrophy. And these are different anatomical regions in the brain. This is true whether you use structural connectivity or functional connectivity to define connectivity. So in other words, areas that are connected tend to show co-atrophy. So that's consistent with the propagating model, but it doesn't prove it, because areas that are connected also tend to share properties. They tend to express the same proteins, they tend to have the same cellular distributions. So it may just be that those areas are equally vulnerable to the disease. And that's a theory that was proposed really, as I said, until a few years ago. So how do you test the prion model? So one idea, so this is basically a summary of the prion model in Parkinson's disease. So alpha-synuclein becomes misfolded. In its misfolded state, it can cause other proteins to misfold. So, if in red you have the misfolded protein, it meets a normal protein, and it has a chaperone-like model of misfolding the other protein. So the normal protein becomes misfolded, then they separate, and then they can both go and misfold another protein. It's almost like an infectious disease spreading through a brain. And so can you test this hypothesis? And so we were inspired. This is actually an idea that was given to us by Yasser Ituria. We were inspired by work in infectious diseases epidemiology. And I just want to show this really interesting slide that was brought to my attention by Albert Getty, a mentor and colleague. Peter Panum was a young medical student, 22 years old at the time, who was sent to the Faroe Islands to go study an epidemic of measles that had broken out in the Faroe Islands. And what was interesting about the Faroe Islands is that it's full of little fishing villages that are not connected to each other. And they're only connected when someone by boat goes from one village to another, which is a very rare event. And so what he realized is that the measles would propagate in one village and then spread to another village when a person went by ship from the first village to the second village. So he concluded that measles, based on observing this temporal spatial distribution, he concluded that measles must be an infectious disease communicated from person to person. And this was not known at the time in the 1850s. He also observed that in the Faroe Islands, there were epidemics of measles, unlike on the continent, and that they occurred every 60 years. And he reasoned that this was because once you had measles, you became immune. And so the epidemic could not occur for another 60 years until you had enough new people who were not immune and who had never had the disease. So he made these incredible observations based only on observing the spatial temporal distribution of the disease. And I'm going to suggest that we can do the same thing with brains. So this is actually the paper that Yasser pointed out to us. It's by Brockman. And it's a model of the spread of infections in the world. Here he's modeling two types of infections with an epicenter either in Mexico or China. And he's using airplane travel as the route of propagation of the infections. And what's really fascinating is that he showed, using what's called an agent-based model, where you model each individual who is infected and their travel, he showed that you could replicate the spread of these diseases using airplane travel as your mode of propagation. He did this also for COVID, did a lot of really fascinating research on COVID here. But the point is that when we read this paper, we were interested in using that same model. It's called an agent-based model. And instead of modeling the spread of an infection via airplane travel, we tried to model the spread of prions through the brain's connectome. So instead of infected individuals, we now have infected proteins, and instead of airline routes, we have brain connectome. And the main person who did this work is Yingqiu Zhang, in collaboration with Bratislav. So she developed this SIR model of propagation of alpha-synuclein. And SIR is an agent-based model in which each agent is susceptible, infected, or recovered. And so what you do in this model, you inject a misfolded protein in a part of the brain, and then you let it propagate through the connectome, and you let it infect other proteins. And then you just let the model run and see what happens, and see if you can predict brain atrophy using this model. So this is an example of using computational modeling to inform mechanisms of disease. So the other thing that we used here is we used the Alain Human Brain Atlas, and we used the genes for synuclein, that's the abnormal protein, and GBA, which is a gene that's involved in the clearance of alpha-synuclein. So in our model, we have spread of the protein through the connectome, but we also have synthesis and degeneration of the protein using these proteins. And the first thing we found is that the model does replicate true brain atrophy. But what's interesting is that we can use null models to see if various parts of the model are important for the fit. And so, for example, here we use the SNCA gene distribution from the Alain, and then we randomly change the distribution of alpha-synuclein. And what you can see is that the true fit is much higher than all the null models. So this is a way to do what Judea-Pearl would call counterfactuals, and to test your model, using, in this case, by randomly shuffling something that you think is important for the model. So what this shows is that the distribution of alpha-synuclein is important for shaping the spread of atrophy, really supporting the prion model. So the last thing I want to show in the last few minutes is how you can use genetics to better understand vulnerability to Parkinson's disease. So Parkinson's disease is mostly sporadic, but even in sporadic cases, it's quite heritable. And there has been a recent GWAS of Parkinson's disease using many tens of thousands of cases and controls. What you can see is that if you have a higher genetic risk of Parkinson's disease, these are quartiles of genetic risk, your risk of developing the disease goes up almost 8-fold. Now, if you're in the highest quartile, you're still not likely to get Parkinson's disease. These people in the lowest quartile may have a 1% chance of developing Parkinson's disease in their life, and these high-risk people only have an 8% chance. So much higher, but still likely not to get the disease. So similar to our essential tremor experiment, we used the UK Biobank, and we used about 40,000 participants, and we computed a polygenic risk score of Parkinson's disease in each individual. And what we predicted was that people with high risk of Parkinson's disease would show subclinical evidence of neurodegeneration. We found the actual opposite. What we found is that people at high risk for Parkinson's disease have widespread increased cortical surface area. So this is measuring the correlation between polygenic risk and surface area. And you see this quite extensive increase in cortical surface area in people at high risk. We also saw increases in all subcortical volumes of all the subcortical nuclei. And we also saw increases in white matter fractional anisotropy, indicative of increased white matter density. So in other words, people at high risk for Parkinson's disease have bigger brains. They have increased cortical surface area, more white matter projections, and increased subcortical volumes. So they don't have subclinical Parkinson's, they have bigger brains. We also find that now in much larger numbers of individuals, over 200,000 from UK Biobank, people at high risk for Parkinson's disease genetically have higher education level, higher household income, higher fluid intelligence, lower BMI, they live in areas with less deprivation, and they're less likely to smoke. In other words, this is very consistent with having the bigger brains behaviorally. So what we suggest from this is that the genetic risk for Parkinson's disease is associated. So I just have a couple more slides. It's only 5:15. So just a couple more slides to finish this story. So genetic risk for Parkinson's disease is associated with larger brains and cognitive features and lifestyle features that are associated with this. And then, the last thing we did to show that this was a true mechanistic relationship, we showed that surface area expansion in Parkinson's disease overlaps with cortical thinning in Parkinson's disease. In other words, areas that show the greatest amount of surface expansion show the greatest cortical thinning. We also did Mendelian randomization, which is a technique to infer causality, to show that actually bigger brain is the cause of Parkinson's disease. So I will wrap it up here and go to my conclusion slide. So I've shown 4 experiments where we can try to infer causality from observational data, inspired by Pearl. So we've shown that either genetics or biologically informed models or both can be added to big data to try to understand causal relationships. And Pearl has this hierarchy of causal inference. The first level is seeing, just doing correlations, including causal graphs like structural equation modeling to account for confounds. The second model he calls interventions or doing. So an intervention might be telling people to quit smoking and then seeing if that reduces the risk of cancer. But another kind of intervention is using genetics as an intervention on your data, as in Mendelian randomization. And then the third level of causal inference he calls imagining, which is basically generating models of what you think is happening in the brain or in a disease, and then testing counterfactuals. So I hope I've convinced you that, that it is possible to infer causation from correlation when you use different approaches. Thank you.